Hello and welcome from my side to today's webinar. My name is Felix Eppler. I'm product manager for cell sorting solutions here at Miltenyi Biotech. And in today's webinar, we would like to address the question whether the common cold coronavirus is either a protector from or rather an exacerbator of COVID-19. Our webinar will be split into two parts today. First, I will talk about a technical highlight, the MACSQuant Tyto cell sorter, a system which allows for aerosol-free and gentle cell sorting in a closed cartridge system. And in the second part, Alexander Scheffold from the University of Kiel will talk about a study he and his group conducted recently and focus on antigen-specific CD4 positive T cell responses in humans and thereby on SARS-CoV-2 and beyond. So let's start with a technical introduction and let's have a closer look to the MACSQuant Tyto cell sorter. But before we do so, I would like to give you a short overview on the general product portfolio at Miltenyi Biotech. As you can see, we try to cover whole workflows, starting with sample preparation, including subsequent cell isolation or cell sorting, cell culture and cell activation and expansion possibilities, but then also cell analysis using flow cytometry as well as microscopy. In an ideal world, these workflows then can be easily translated into the clinical segments, so from benchtop to bedside, and this is also something we are focusing on here at Miltenyi Biotech. Since we're talking about coronaviruses today, I'd also like to take the opportunity to shortly highlight a few of our products which help researchers around the world in order to understand the SARS-CoV-2 in much more detail. These are different reagents, for example, our SARS-CoV-2 Peptivator peptide pools or the SARS-CoV-2 antigens, but also analysis kits and sorting kits, for example, for SARS-CoV-2 specific B and T cells. And of course, there's specific instrumentation supporting these researchers. And one of them, as said before, is the MACSQuant Tyto cell sorter, which I will focus on in the following few slides. So if we talk about flow cytometry-based cell sorting, most of the people think about FACS droplet sorters. These are the systems which you mainly find in the laboratories and core facilities. If we talk about sorting of infectious material, these systems come with a few problems. And that is the first, that they are open system and produce droplets as well as aerosols in an open space. But they also share their fluidics and tubings. And for most of the systems, it's very difficult to exchange those. And this means very difficult and time-consuming decontamination processes are needed in between samples. Last but not least, if you're thinking about going into delicate downstream analysis of the cells, for example, single cell genomics, but also functional analysis, a problem can also be that the sorting process itself on droplet sorters can be very harsh and depending on your cell type, create problems with downstream functionality or also artifacts in your analysis. And we think that we have a very good alternative to conventional droplet-based systems with the MACSQuant Tyto cell sorter. The MACSQuant Tyto is a benchtop-sized cell sorter allowing for a controlled sample temperature between 4 and 25 degrees Celsius. And there are no external fluidics. So really what you place into your laboratory is what you can see here on this picture. The Tyto comes with 3 different lasers, 8 fluorescence channels and 4 scatter channels, and thereby allows for quite some flexibility in terms of antibody panel design. The heart of the system is the MACSQuant Tyto cartridge, which has 3 different chambers. An input chamber, where the sample of interest goes into, a positive collection chamber, where the target cells will end up after the sorting procedure, and a negative collection chamber, where all the rest will simply flow into. Cell sorting as well as analysis take place at a small microchip at the bottom of the cartridge, which is depicted in this picture here. Just for your understanding how small the microchip really is, I'd like to show you this size comparison here. So in order to understand its architecture a little bit better, we need to zoom in. And here you can already see the channels which directly connect the microchip with the different chambers of the cartridge. If we zoom in again, we see the architecture of the chip. You can see different channels, and the cells coming from the input chamber enter the microchip at the lower left-hand side and then run through the sorting channel, where the cells are analyzed. If a target cell is identified, the sorting procedure will be initiated, and the sorting procedure itself will be carried out with a very fast microvalve. We can also have a closer look to this procedure. So here you can see the microvalve, which is actually opened by a magnetic pulse coming from a solenoid. One opening and closing cycle takes around 30 microseconds, which allows for up to 30,000 microvalve openings and closings per second. You can see an animation of the sorting mechanism itself. Cells coming from the input chamber pass the 3 different lasers, and in case a positive target cell has been identified, a magnetic pulse opens the microvalve, which then very gently redirects the cells into another channel, leading into the positive collection chamber. All the rest flows through into the negative collection chamber. The only thing coming in and getting out of the cartridge during the sorting process is sterile filtered air. It enters the cartridge at its bottom through an 0.1 micrometer hydrophobic filter, and then, before entering the input chamber, runs again through another 0.1 micrometer hydrophobic filter, ensuring sterility of the air before entering the input chamber. Also, the ventilation ports at the positive as well as the negative collection chambers are covered with those hydrophobic 0.1 micrometer filters. An interesting study coming from different groups at the National Institute of Health in USA, which has been recently published in Applied Biosafety, validated the application of the MACSQuant Tyto cell sorter in a BSL-3 containment setting. So what did the authors do in this study? They used fluorescent beads and sorted them either on the MACSQuant Tyto or on a BDFAX Area 2 with the aerosol management system disabled, and then used the gold standard for detection of aerosolization of instruments, Cyclex D impactors, in order to check whether aerosols and thereby also fluorescent beads had been released from the systems during the sorting process. As you can see on the right-hand side, the FAX Area 2 with its aerosol management system disabled released a lot of fluorescent beads, which is a strong indicator for strong aerosolization during the sorting process. In contrast, the MACSQuant Tyto did not release any aerosols at all during the sorting process, also not once the cartridge was opened post-sorting. The authors concluded that this instrument is not only suitable for BSL-3 due to its small footprint, but most importantly because it does not generate aerosols during the sorting procedure. So let's shortly sum up. Where are the benefits of using the MACSQuant Tyto for sorting of infectious material? We have a closed and sterile sorting environment and no sample-to-sample carryover, as there are no internal fluidics or tubings present in the system. If you use a new cartridge, you always use a completely new fluidic system. There are also no contamination and therefore also no decontamination processes needed in between samples. And most importantly, no aerosols are produced with a microvalve-mediated sorting process. But it's not only about safety, it's also about the gentleness of the MACSQuant Tyto. It operates at very low pressure, and therefore the cells do not experience any decompression or charge, and also only encounter mild shear forces, which is of course very important for delicate downstream analysis of functional assays which are conducted post-sorting. One of these delicate downstream analysis, which is in a strong need for high quality samples and therefore also for gentle cell sorting technologies or in general gentle sample preparation technologies, is the genomics analysis of cells. For example, if you're interested in transcriptomics. As you can see on the right-hand side, in a direct comparison between epithelial cells and their transcriptomics after they had been sorted either using the MACSQuant Tyto or a conventional droplet sorter, you can see that certain signaling pathways directly connected to stress, such as apoptosis, mitochondrial genes, or oxidative stress, that these are higher and stronger expressed in the cells which had been sorted using the droplet sorter. So it really is key what kind of technology you use, and it will influence the data you will receive in the end. If you're interested in more details on this application and the background of the study, then please have a look at another webinar from our site, Realize Better NGS Results Through Gentle Cell Sorting. This is available at the Miltenyi Biotech website as well as here on LabRoots. And with that, I'm really happy to hand over to Professor Dr. Alexander Scheffold. As said, he's the director at the Institute of Immunology at the Kiel University in Germany. And today he'll discuss the antigen-specific CD4 T cell response of SARS-CoV-2 patients and their connection to the cold coronavirus. And with that, I'm already looking forward to listen to your talk, Alexander. And yeah, thank you for your attention.
Welcome to my talk about SARS-CoV-2 specific CD4 T cell responses in healthy donors and COVID-19. My name is Alexander Scheffold and I'm the head of the Institute of Immunology at the Christian Albrechts University in Kiel. So we still don't understand many aspects of COVID-19. And for example, one striking question is, why do some people develop severe COVID-19 whereas others remain, for example, completely asymptomatic? So there is the idea that adaptive immune responses play a key role in that process. So they could be too little or too strong or just inappropriate responses. On the other hand, there's the question whether pre-existing immune memory could have an influence, either protective but maybe even harmful. So the characterization of SARS-CoV-2 specific T helper cells, which are actually orchestrating the adaptive immune response, is one of the key aspects of analyzing the adaptive immunity. So one can look on the frequencies, on the phenotypes, also on the antigenic targets, but also looking on the functionality of these cells, such as TCR specificities and cross-reactivities, but also the TCR affinities and clonality. Antigen-specific T cells can be identified in two different ways. So either by direct staining with a ligand, this is the peptide MHC molecule, and this allows direct labeling of the antigen-specific T cell receptors, and then these labeled cells can be identified via conventional flow cytometry. This is the only way of direct TCR labeling, but it requires that we have exactly defined MHC and peptide complexes which need to be prepared. And so far, the main analysis is still done in CD8 T cells. The classical way of detecting T cell reactivity is via functional assays. So the idea is just to add the antigen to a suspension of antigen-presenting cells together with T cells. The APC take up the antigen, process the antigen, and present the peptides on the MHC molecules. And then the antigen-specific T cells get activated and start to react with a defined set of effector functions, including rapid cytokine production, expression of certain so-called activation markers on the cell surface, or after a longer time of simulation, proliferation occurs. In our hands, the activation markers CD154 and CD137 have been particularly useful because they require minimal manipulation of only 7 hours in vitro restimulation. They can be used to detect basically all pathogens and antigens, ranging from peptide pools, antigen lysates, or recombinant proteins. Of course, this is possible in all donors because we have no MHC restriction. And all T helper subsets somehow upregulate one of these markers, including the FOXP3 positive regulatory T cells. So for some general questions regarding antigen-specific cytometry, I would like to recommend this article in the European Journal of Immunology. This is a general guidelines for flow cytometry, covering basically everything you can do with flow cytometry and cell sorting. And this also includes an overview about the different technologies which can be used to identify antigen-specific B and T cells via cytometry. And the technical aspect in that case has a considerable impact on the interpretation of the data actually. So one general problem of antigen-specific cytometry is that these cells are typically very rare. And here I just show a couple of examples. This is a CD154-based stimulation-based assay detecting CD154 on reactive cells. So this is very low background without stimulation. And here are some classical pathogens. And you can already see that typically the frequency of cells is way below 1%, in some cases even below 0.1%. And that is summarized below here in that cartoon, where you can see that the frequencies are really 1 in 1,000 or even less in the case of autoantigens or naive T cell repertoire. So the technology we used for identification of antigen-specific T cells is also based on this in vitro activation with antigen for 5 to 7 hours. And that leads to upregulation of these two molecules on a cell surface of activated T cells. Regulatory T cells make CD137, conventional T cells make CD154, the CD4 T ligand. And here you can directly see the frequencies are really low, about 0.1 or 0.2%. So what we did, we combined this detection technology with a magnetic filtration step, allowing to separate the rare reactive cells from a large population of cells, 10 to hundreds of millions of PBMCs, and we can filter out the few reactive cells, and this gives then a sizable population of regulatory T cells, shown here in blue, the conventional T cells, and one can now do a really in-depth analysis of these rare reactive cells, looking also on the quality, so on the phenotype, the function, the TCR affinity, cytokine production, and do also all kinds of omics technologies due to the purification of these cells. So when we applied this technology to blood samples of COVID-19 patients, we got quite striking responses as shown here in COVID-19. So the main reactivity was directed against the classical targets spike membrane and M protein, but also some reactivities against other proteins were identified. Interestingly, when we looked at unexposed donors, we also saw basically in every patient, in every donor, we found SARS-CoV-2 reactive T cells also distributed against various antigens. And this is summarized here, showing that although there's a clear-cut difference in the frequencies of SARS-CoV-2 reactive T cells in COVID-19 versus healthy unexposed, basically in every donor we were still able to identify the SARS-CoV-2 reactive T cells. And that is just influenza as a control. There's no difference between patients and healthy. One can summarize that. So the reddish labels is indicating the three main targets, spike, M, and N protein. And these are the main targets, especially in COVID-19 patients, in infected patients. Whereas in unexposed, it's more scattered. There are considerable variabilities. So in some donors, also other proteins are the major targets. So to summarize that, S, M, and N protein are the main targets in COVID-19. There are more diverse targets in unexposed donors, but in general, pre-existing memory T cells can be detected in all unexposed donors. So now, of course, the question is, are these really SARS-CoV-2 specific T cells we are detecting here? And this is now showing the original data. This is after stimulation before enrichment. There's a low frequency of reactive cells in a COVID-19 patient, and this now shows the dramatic enrichment of a lot of reactive cells by magnetic cell sorting. And the same is actually true in unexposed donors, although we can clearly see that here we have much less reactive cells, but still we get a considerable enrichment of these cells from the unexposed donor. So are these specific? We tested this by generating in vitro expanded T cell lines from these enriched cells, and then re-stimulated them with the SARS-CoV-2 or control antigens. And as you can beautifully see in COVID-19 as well as in unexposed donors, These T cell lines exclusively react to the inducing antigen, but not the control antigen. As you can already see here, in the unexposed donors, there's this kind of a higher background reactivity against the other antigens as compared to COVID-19, where the background is almost zero. I will come back to that later on. So we were wondering about this pre-existing T-cell immunity, and there was the hypothesis that this might be induced by cross-reactivity to common cold coronaviruses, which are causing the common cold in normal people. And the idea was that maybe children, because they are frequently infected with these common cold viruses, are better protected from SARS-CoV-2 as compared to elderly, because some of these related antigens cause cross-reactive protective immunity. So as I said, we can detect these antigen-specific cells in all unexposed donors. But when we looked at the phenotype, there was a clear-cut difference. So whereas in COVID-19 patients, there were no naive T cells, as indicated here by CD45RA and CCR7 expression, as expected for a memory in vivo memory T cell response. However, in unexposed donors, these SARS-CoV-2 specific cells, there was a considerable fraction of naive T cells. And that was actually true in all unexposed donors. So it's a highly variable amount of memory versus naive T cells in these unexposed donors against basically all three main target proteins. And again, the control influenza, as you expect for a memory response, they are mainly comprised of memory cells in unexposed as well as in COVID-19 patients. And most strikingly, again, COVID-19 patients, the SARS-CoV-2 specific T cells were almost completely to almost 100% of a memory phenotype. So this already suggested that there's a significant heterogeneity of memory and naive T cells. So the question is, what is the reason for that? Is that due to cross-reactivity, or is that a general phenomenon within the total T cell subsets? And so we just just want to show some data on the total CD4 repertoire, because as we know, the aging has a significant impact on immune repertoire. And that is shown here for two examples. This is a young donor and an old donor. And this is just looking here on CD45RA, CCR7 again. So we see in the young donor, we have a 70% of naive T cells, whereas in the old donor, there are just 7% of naive T cells. And now, interestingly, when we now enrich the SARS-CoV-2 specific T cells, and these are both unexposed donors. And when we now analyze these SARS-CoV-2 reactive T cells, we realize that also here, the young donor, 70% of the SARS-CoV-2 specific T cells were also naive, versus in the old donor, there were almost no naive T cells in the SARS-CoV-2 reactive cells. And we can plot that, and that is indicating actually that it is just a linear, almost linear correlation between the total frequency of memory cells within the CD4 compartment, which we call the immunological age or the immunological experience, increasing memory frequencies. And this is linearly correlated with the frequency of memory cells in the SARS-CoV-2 specific T cells. So indicating that this is not due to a particular pre-infection with a particular virus, but is more or less a function of the general broad memory T cell repertoire, which is increasing in healthy people via aging. And that is actually not restricted to SARS-CoV-2. We see exactly the same linear correlation in CMV negative donors against also the CMV specific T cells have exactly the same shape. And again, against a complete neoantigen, the keyhole limpet hemocyanins, KLH, we see the same effect. And again, when we look then as a comparison in CMV exposed or SARS-CoV-2 exposed donors, there's clear-cut a pure memory phenotype due to the in vivo activation and expansion and memory formation due to the in vivo infection. So this is actually indicating that the SARS-CoV-2 pre-existing memory correlates simply with the size of the total memory pool, which we call also the immunological age. And now the question is, do these pre-existing memory cells differ from the memory cells we found in the COVID-19 patients? So what the next we did, we looked on the functional avidity of these cells. So we took specific T cell lines generated from unexposed or in COVID-19 patients and re-stimulated these cells with graded peptide doses. And then, as you can nicely see, in unexposed donors, you need extremely high concentration, up to 0.5 microgram per ml to get a response. Whereas in COVID-19 patients, the T cells react already at the lowest dose, 0.001 microgram of peptide. And you can calculate from that the EC50 values. And as you can see, although not all COVID-19 patients develop high affinity responses, but at least some of them do. Whereas in healthy people, so the unexposed, the pre-existing memory cells, they are all exclusively of low avidity responses. So to summarize that, the pre-existing SARS-CoV-2 memory T cells are present in all unexposed individuals analyzed. They have low avidity for SARS-CoV-2. And basically this frequency of pre-existing memory correlates simply with the size of the total memory pool, which is the immunological age or the immunological experience. So the next important question was now, but still, do we see some induction of this cross-reactivity by common cold coronavirus? So we analyzed also the response to the 4 known members of these common cold coronavirus strains. And what we found is that all tested donors had extremely high frequencies of reactive cells, and they were of a complete memory phenotype, indicating really in vivo selection, expansion, and memory formation against these viruses. And that is in striking contrast to the SARS-CoV-2 response in these very same donors. And when they then had a closer look, as expected for a real infection, the common cold coronavirus specific T cells were all of a relatively high avidity, again in contrast to the SARS-CoV-2 of these unexposed donors. And finally, we also had the chance to look here on the cross-reactivity. So we took the common cold coronavirus specific T cell lines and stimulated them either with the inducing antigen, and we got higher reactivities, or we re-stimulated with the SARS-CoV-2, and there was almost no reactivity detectable. So basically, what we found is that we find high avidity and expanded memory T cells against the common cold coronavirus in virtually all tested donors. However, almost no cross-reactivity between these specific T cells and the SARS-CoV-2 antigen. The next was then to check whether these SARS-CoV-2 reactive T cells contain some cross-reactivity. And that is, as first we looked here on the unexposed donors and doing exactly the same experiment, re-stimulation of SARS-CoV-2 specific T cells with different types of antigens. They have high reactivity against the 3 different antigens we used for the generation. However, they have also broad cross-reactivity against a number of different antigens, but that is not only restricted to the common cold coronavirus, but it's also seen against other viruses like influenza, CMV, EBV, and adenovirus. So indicating that this cross-reactivity is not restricted to a particular antigen or virus. And the next was then to look in COVID-19 patients, and the result was quite striking, whereas we still see high reactivities against the 3 main targets. Basically, these cross-reactive cells are hardly detectable in COVID-19 patients, indicating that upon in vivo infection, there's a positive selection of SARS-CoV-2 specific high avidity cells and a negative selection for these low avidity cross-reactive cells. And that can be quantitated by looking on the percent of cross-reactivity in each T cell line. And it is clear that it's very high in the unexposed, but it's almost completely lost in the COVID-19 situation, showing this clear-cut evidence for selection of high avidity monospecific cells during the COVID-19 infection. So the human coronavirus cross-reactive cells, although they are present, they do not really contribute to the SARS-CoV-2 response in COVID-19 infection. And that was actually beautifully confirmed in a recent study where they really analyzed or identified T-cell receptors which cross-reacted especially against a homologous peptide between SARS-CoV-2 and coronavirus, which is frequently identified in unexposed donors. And this work here cloned cross-reactive TCRs and showed that these cloned TCRs really have low avidity, low proliferative capacity against SARS-CoV-2 as compared to the original common cold coronavirus peptide. So this nicely confirms that the pre-existing common cold coronavirus cross-reactive memory cells poorly react against the SARS-CoV-2 protein and therefore are probably not relevant or are at least not protective in a situation of SARS-CoV-2 infection. And I just here want to point out that this antigen-specific cytometry is really helpful also to improve single cell or in general omics data, which are frequently used nowadays to look on the whole blood, however, and also do TCR sequencing on that level. However, we should not forget that so far it is not possible to extract from the TCR sequence the specificity. So by combination of global single cell sequencing together with sequencing of individual sorted antigen-specific B or T cells and identification of these TCRs in the antigen-specific subsets, one can add the specificity and the function into these global single-cell RNA profiling experiments. And that is actually what we did. So we identified the antigen-specific T cells in SARS-CoV-2 in COVID-19 patients. We did the ALTE technology, and then we made use here of this beautiful closed system with the Genomics Tyto and sorted these reactive cells to really high purity, which allowed us then to do functional assays, but also single-cell RNA profiling. And the idea was here to look whether we see differences between patients with mild disease versus severe disease. And the first striking observation was when we looked on moderate and severe COVID-19 patients, we found that those actually have strongly increased frequency of reactive cells. So it's not an impaired immune reactivity in the cells as it was previously reported with other methods. However, the really striking result came when we also now looked on the avidity of these T-cell responses. And what we found is that in non-hospitalized patients, they had a strikingly high avidity response, suggesting a good selection of high affinity TCRs during infection. Whereas in a hospitalized patients, the avidity was similar to the unexposed, indicating that under these conditions, although the response is very strong, the T cells are suboptimal because they only react to SARS-CoV-2 with a very low affinity. And then we also did single cell sequencing and identified the TCR profiles of these ex vivo isolated SARS-CoV-2 T cells to look on the clonality of the response. And what we can see here that in hospitalized patients, there's a very high diversity indicating that there is no clonal or only weak clonal selection upon infection, whereas in non-hospitalized patients, the diversity was much lower, indicating really a selection of individual high affinity clones. And that is also somehow reflected here when we look on the different subpopulations within the antigen-reactive T cells. And we here just look on in black, the top 3 expanded clones. And you can see here that they are all collected here in one particular subset, which is actually characterized by higher activity. Whereas in severe disease, there is not such an accumulation, so they are scattered all over the place and found in all different subsets which were identified in this experiment. So this is actually indicating that this is a strong but an unfocused and low avidity CD4 T cell response in severe COVID-19. And now, of course, the question is, does the pre-existing memory, which has also this low affinity, contribute to such a low avidity response in severe COVID-19 patients? And we could not completely address this because we had no samples before and after infection. However, we tried to filter that out of our data by looking only on a patient group with the age between 50 and 65. And when we looked on them, we still see the same phenomenon. Severe patients have still high frequencies of antigen-reactive cells. However, it was interesting to observe when we now look on the immunological age of these patients. So that's just looking on the frequency of memory cells in the total CD4 cells, we see that the severe patients have much higher immunological experience or age as compared to the non-hospitalized, versus in healthy, there is a classical broad distribution of all memory frequencies. So this is at least suggesting that patients with severe disease have a higher immunological age, and this might actually contribute to the development of a poor antigen-specific response upon infection. So to summarize that, increased frequencies of SARS-CoV-2 specific T cells are found in severe COVID-19, and this is independent of age. The increased immunological age correlates with COVID-19 severity in this age group. And we still have to do analysis of paired samples before and after vaccination. That is now much more easy than with natural infection that is ongoing. But I just want to show you a couple of, or the first results on vaccination. So that is just showing the frequencies. So pre-vaccination level. And we see a very strong induction of SARS-CoV-2 reactive T cells upon 2 times vaccination. And now again, the interesting part is again the young and old donor I showed you before. So the one which has this large fraction of naive T cells in the SARS-CoV-2 reactive T cells versus the old donor who has almost no naive T cells in the SARS-CoV-2 before vaccination. And if you now look just on these 2 donors on the response, you see that the young donor had a very low starting level but has a dramatic increase of reactive T cells, whereas the old donor has a higher starting level of memory cells but has only a very mild increase upon vaccination, suggesting that the older patient reacts poorly against the vaccination. And that is actually true for a number of donors as shown here. So there's a beautiful negative linear correlation of the more memory T cells we have pre-vaccination, the lower is the fold expansion in response to vaccination. And the same can be done with just looking at the frequency of memory cells in the total CD4 cells, so the immunological age. And that also has a negative linear correlation with the fold expansion, indicating that the more memory cells we have against SARS-CoV-2 or in total, the lower is the reaction to the vaccine. So to summarize that, so I wanted to make the point that the analysis of the rare antigen-specific lymphocytes is really an important tool to better understand protective and also pathogenic immune responses. And you should not forget that it's just much less than 1% of all CD4 T cells, but they make the difference in such antigen-specific diseases. And pre-existing SARS-CoV-2 reactive memory T cells are really present in all humans, but they have only low functional avidity and they have broad target specificities. On the other hand, human coronavirus, so the common cold coronavirus, they are also very abundant, but they do not strongly contribute to the SARS-CoV-2 response following infection. And finally, the most interesting result probably is that in severe COVID-19 patients, we see a very strong response, but this is of very poor quality, indicated by low avidity and polyclonality against SARS-CoV-2. We can also show that the expanded pre-existing memory correlates with a weak response to vaccination. So in general, the last comment to pre-existing or cross-reactive memory and human T-cell responses and aging. So it seems that there is a correlation of the size of the CD4 memory compartment, which is somehow related to the immunological age. And this seems to be a risk factor to develop severe COVID-19. This we still have to show, but we have already indication that it correlates with a weak response to vaccination. And that will be analyzed in the future by our group. So as a last point, I want to thank the people involved. So the major contribution is by Petra Bacher. She was mainly responsible for planning and organizing this study together with members of her group, Gabriela Rios Martini, together with members of my own group. The IKMB in Kiel doing the single-cell sequencing analysis. And then we have a lot of clinical cooperation partners, especially the people from the University Hospital in Cologne and also in the hospital in Frankfurt, who contributed a lot of clinical samples necessary for doing this analysis. Thank you very much.
Yeah, and also from my side, thank you very much